My name is Owen Witte, and I'm the director of the Broad Stem Cell Research Center, and it's my pleasure to welcome you and thank you for coming this evening. I'm really thrilled with the turnout, and it's really nice to see some people who have already been friends of the Stem Cell Center coming back and wanting to learn more about what we're doing, and a lot of new people as well. The purpose of this evening is really very simple. We want to show our progress in applying the science of stem cell biology to important clinical problems that affect all of us and our families. And I want to tell you a little bit about the history of our stem cell center so you can understand where work like this comes from and perhaps where we'll be going in the future. In 2004, passed by a vote of the citizens of California, an institution called CIRM, C-I-R-M, California Institute of Regenerative Medicine, was created through Proposition 71. You'll hear more in a minute from the head of that organization. This was an amazing thing, really, publicly supporting research in a specific area of biomedicine. Really, it had never been done quite this way previously. Funding from this organization has provided a wide range of opportunities for scientists here at UCLA and many other institutions throughout California. The work goes from the very basic, understanding how cells work, how they use their molecular machinery to create products that we can use therapeutically, all the way up to translational events in which we use cells as therapeutic products. At UCLA and our Stem Cell Center have been incredibly successful in competing for these CIRM funds. I'm very proud to say that we have over 65 awards to date here at UCLA from CIRM. It has brought in over $200 million in new research funding to our UCLA campus. And it supported a variety of things, including important disease team grants, a couple of which you'll see exemplified tonight, in which we actually utilize these research monies to test new therapies in the clinic and develop new ideas about human disease as they occur. So there's still some people standing, but there's still some seats if you want to wander down. It's fine. So in addition to these disease team grants, we've also gotten a major construction grant from CIRM to help outfit a floor of a new building, the Terasaki Life Sciences building, some of you may be familiar with. And they've also provided important training grants to build careers of young scientists from graduate school through postdoctoral training, clinical fellowship, and then joining the faculty. Another very important thing in the progress of science is the continuity and training of young scientists. Now, our campus has also been highly committed to this, and when we founded the Stem Cell Center in 2005, then Chancellor Carnicel provided us with $20 million in, I won't say a check, and not cash, but in-kind services. And these are important things like new faculty positions to help build our faculty expertise in this area. It was supposed to provide funding and support for 12 new faculty, but by partnering with multiple departments in the School of Medicine, the college, and across campus, School of Engineering as well, we've actually been able to hire 27 new faculty members covering 11 different departments. And it's this collaborative interaction which I think has made us very successful in most of our endeavors. So we now have over 200 members of our Stem Cell Center campus-wide. They cover every department imaginable, including people from the School of Law, Engineering, and the Hard Sciences. By drawing across the campus, what we've been able to do is things we couldn't do alone. And I think it's that spirit of collaboration that is really our driving philosophy. Another major event for our center was in 2007. Elaine Broad provided a gift of $20 million to match the university's commitment. Now, most times when a private philanthropist makes a gift like that, it usually goes onto a building or some sort of capital construction. But discussions with Mr. Broad, he asked me what would make the greatest impact. And I said, not another building at this point. We actually had a lot of buildings in progress at the time, but rather money to support young scientists and people with bold ideas. And that's what we've put that money towards with great effect. And although the Broads can't be here tonight, I just want to publicly acknowledge the incredible impact that their gift has made and the grateful appreciation that we have for it. Now, the story about the Stem Cell Center, it is sometimes about money, it is sometimes about new buildings, and it is sometimes about specific research projects. But the story for me is the way that our students, faculty, and staff have really come together to create an intellectual environment and this common purpose of taking our science and driving it all the way to the clinic to make a difference in the lives of patients and families who suffer from diseases. Tonight, I think you'll hear some wonderful examples of that, and I think you will become certainly aware that UCLA is one of the leading centers for stem cell research in the country. Now, thank you very much for that, and I now need to introduce a very important person, which is John Thomas. John Thomas is the chair of the governing board of the California Institute of Regenerative Medicine, which is providing this $3 billion in funding over a 10-year period for this area of science. He's a local to Southern California. You can read more about him in the handouts that we've provided. He's a co-founding partner at Saybrook Capital, which is an investment banking and private equity firm, which is right here in Santa Monica. He's had a lot of experience in the biotech world, investing in biotech, so he understands the impact of how new science and technology can bring things forward, and was in the early round of financing of a company called Advanced Cell Technology, which recently received Food and Drug Administration approval for 2 embryonic stem cell-based clinical trials, an important step forward in this field. He also has a very broad knowledge of governmental agencies and a long-standing commitment to patient advocacy. So it's my pleasure to introduce Jonathan Thomas.
Thank you very much, Owen, and good evening, everybody. I want to first congratulate UCLA. I'm going to say this several times, but one of the first things I want to say is we at CIRM have put a lot of time into funding transformative research throughout the state, and this is the first event that I've seen where somebody who's got extensive stem cell research going on has convened a meeting to talk to the community to tell them what's going on. This stuff is super exciting, and I just think this was a great idea. So Owen, I congratulate you on doing this, and I assure you I'm going to be taking this idea to our other funders at various places throughout the state so that they too can learn about the very exciting research going on. This is a strange time for medical research. All of you know, I'm sure, that NIH, which for many, many years was the principal provider of funding for research nationwide, is in a very messed up state due to the fact that our Congress and those back in D.C. cannot get their act together and understand the significance of medical research. They're, in my opinion, very short-sighted in worrying about budgets today and not understanding that if we don't do something about the spiraling healthcare costs that are going to be a major problem and increasing over time, Medicare and federally related expenses coming from healthcare issues will bankrupt this country if we don't do something about it. So there are a lot of issues with NIH right now. Not only is the funding at best level or at worst in the sequester getting cut back year after year, we're now seeing a domino effect where scientists who are doing research all over the country cannot get funding and are threatening basically to leave to go to other countries to be funded and will be doing research there. Big, big problem. It's within that context that CIRM operates, and CIRM is a very interesting beast. As Owen suggests, it was created by Prop 71, a ballot measure passed by the people of California to change the paradigm of how to fund medical research, and that is exactly what it's doing. Interestingly enough, it's an insulated entity which not only cannot be touched or affected by the federal government, it really is insulated from the state government, even though we are a state agency. We were authorized to spend $3 billion on medical research, and the state cannot take any of that money. It's a right written into the California Constitution to do stem cell research and to provide the funding for us to do what we do, which is basically to turn California into the epicenter of regenerative medicine research worldwide. I can report to you that we are the envy of researchers everywhere because we have an unparalleled source of funding to put out for this transformative research. Lots of really neat stuff going on. There are terms you may have heard about in the press. I'm sure friends Don and Owen, etc., will tell you more about tonight. Things like reverse engineered skin cells that they can take back to embryonic form and reprogram to become different types of cells in the body that be subjected to all sorts of research. Embryonic stem cells, which you've heard about, which are the gold standard from which everything in your body has emanated. There are adult stem cells. Your skin is made up of stem cells. Every time your skin sloughs, it is regenerated through stem cells in your skin that make new skin cells. There are things called cancer stem cells, which are thought to be responsible for the proliferation of cancer and contribute to the fact that cells proliferate unimpeded by the immune system, which is thought basically to not be able to see the cancer as it proliferates. You've heard things in the press recently about cloning. That is a topic that happened with Dolly the sheep many years ago, is now resurfaced. All of these things are happening, and in medical terms are happening in rapid fire, hugely accelerated pace, and that's largely in California because of the white-hot scientists like you have here at UCLA, and the fact that we're fortunate enough to be able to fund what they do and to allow them to have facilities, have the infrastructure, have the training programs, have everything to be able to work on their research and to come up with the great stuff of the kind you will hear about tonight. UCLA is a huge part of our program. They're the #2 funded entity out of almost 60 in the state. Between academic and research institutions and biotech companies, UCLA is #2 behind Stanford and has been at it for less time than Stanford has, as Owen is fond of pointing out, and undoubtedly UCLA will lap Stanford, right, Owen? Is that correct? Yes, that's correct. Better than you did in football over the weekend, yeah. It's all right, SC lost too, so it's... unfortunately, so did anyway, that's another story. The programs they have here span the research spectrum. So you can have the very beginning of research is basic research. We have a lot of that going on at UCLA, and that is what ends up developing down the road into what become human clinical trials ultimately. And UCLA has everything in between, from the basic research to the next step, which we call early translation, to preclinical trials, which just means work with animals, and then human clinical trials, culminating in, Owen referred to disease teams. We have 2 of them here that Owen and Tony are going to be speaking about, and Don, I guess we have 3, Owen, correct? We have 3? 3, yeah. And these are $20 million awards. That is a huge number for medical research that we're fortunate enough, because of your foresight as voters, to be able to make available here. There's a huge emphasis on education here. There's a great training program. There was a glowing quote by the reviewers who were evaluating whether to fund UCLA's training program about the environment and the mentors and the facilities, et cetera, which I would read would make Owen blush, so I'm going to pass. Just to tell you, there's tremendous training going on here and a pipeline of young researchers that are going to be doing big stuff down the road. We also have an auxiliary program in the educational space where we partner places that don't have labs in stem cell research. We call it the Bridges program. And here, Cal State Northridge sends students to work with the scientists at UCLA to develop their skills and further increase the pipeline of scientists down the road. So a lot of great stuff going on at UCLA. We are tremendously impressed at CIRM, and we are, Owen and Don and Tony, not to put a lot of pressure on you guys, we're looking for big stuff down the road. 10 to 20 years from now, when the annals of medical history are written for this era, you mark our words, you will look back and this will have been one of the golden eras of medical research. And gentlemen and like these and the teams they have are directly going to be responsible for that. And you will have made it happen, which is a very cool thing to do. So on behalf of the citizens of California, the state of California, the researchers, we thank you and welcome you to a most interesting evening.
Thank you for those lovely comments. I'm going to have to invite you over more often, I think. So it's my pleasure to now introduce Dan Geschwind. Dan is our first speaker of the evening, and he's really a world-renowned neurologist. He holds an endowed chair in the School of Medicine. He has appointments in both neurology, psychiatry, and human genetics, and is director of the Center for Autism Research and Treatment. He sees patients, he does basic research, he connects it all, and I think it's really the type of person that exemplifies what CIRM and the funding of the Stem Cell Center is all about, is that he made a proposal to a grant program that we run through the Stem Cell Center called the Innovation Program. And it was a little bit, I'd say, almost on the wild side, but yet he had such a passion for it and such an idea to drive it home that we funded portions of the research he'll be talking about tonight, in which he's going to be able to try to model this very complex disease of autism as a disease in a dish. And it's a strategy that many have used, but I think it's really being done at the absolute highest level here. So please, Dan Geschwind.
Thank you. Thanks, Owen. It's really a great pleasure to be here and to talk about this work that started because of this Innovation Pilot Award. And my title, Moving from Genes to Therapies Using Stem Cell Models. What I'm just going to do is try to fly over this at about 25 or 30,000 feet to give you an introduction as to what we're doing and why we're actually trying to do this and how one could possibly do it. So our model in our laboratory is to identify genes that cause diseases and then use those to get a mechanistic understanding of disease. You can understand the cause, the etiology, by understanding the genetic basis. And it turns out that autism is not 100% genetic, but has a high genetic liability. In fact, and I'll discuss that in a little bit. But essentially, what we're dealing with are diseases of the brain. And at some level, the genetics is a toolbox that we can use to understand how the brain develops, the different brain regions, and how they might connect, both at a cellular level and at a circuit level. And you may not be able to see this that well in this auditorium, but again, we're talking about studying cells here. And essentially, the DNA goes to something called RNA that makes the protein. It's as if the DNA is a blueprint and the protein is the factory or the machines in the factory, and the RNA is in between that. And a lot of what we're doing is studying this RNA, which is a readout of what's going on in the cell, to give us a kind of fingerprint of what's going on. And I'll discuss that just very briefly. But that's the notion here, that we can go all the way from this to cells and then to brain function in some way. And that's the challenge, and that's what our lab's been working on in several neurologic diseases. And here I'm going to talk about autism, which has been something we focused on very heavily for the last 10 years. And really, again, this is a classic model that you've probably seen before, perhaps even a newspaper. Find genes. You can use genetic engineering to make those animal models or mice that carry those mutations. These in vitro human models using stem cells. We can even study postmortem tissue. We need to put all this together to develop therapeutics, which is our goal. So the notion is we're not a mouse lab, we're not a stem cell lab. We're a disease-focused lab, and the stem cells are a new critical tool in our armamentarium to study the disease. And again, another kind of approach that we use in our laboratory is a computational, you may have heard the word systems biology, but it's a way of, instead of studying one thing at a time, we're trying to integrate multiple levels of data, going all the way from the genes to the phenotypes. And for those of you who haven't heard genetic talks, the phenotype is anything you can measure. Here it would be the autism. As my oldest son used to say when I'd come home from work, he goes, "Dad, how are the phenotypes today?" And so he got what that is. And so I work, as geneticists, we work on phenotypes. So autism is a kind of one of the most fundamental neurodevelopmental disorders. I think of it as prototypical because it involves so much of development between age 0 and 3. Language and social brain, repetitive behaviors, and it has a high overlap with intellectual disability. About a third of the patients have severe intellectual disability. Also about 10% have epilepsy. So these diseases have an overlap, and part of the job is to find the genes and understand how they could cause something like autism and intellectual disability and epilepsy altogether as well as separately. And what this is basically showing is these are pictograms of the human chromosomes, and you can see every single one is colored on. And the coloring basically tells us that there's a gene for autism that's been found there. So pretty much there's a gene or multiple genes that have been found all over the genome. It's a very heterogeneous disorder. There is not one gene causing autism. In fact, advances in genetics have led us to appreciate that there are probably about 500 to 1,000 genes that cause autism. None of them, even major effect genes that themselves can cause autism in a child, do not account for more than 1%. So if we have 100 children with autism, we might have 100 forms. It's very analogous to cancer, or even cancers that might look the same, might be very heterogeneous underneath in terms of the kinds of mutations. So this poses a lot of challenge. And so as scientists, we need to begin to pick this apart gene by gene. But what we're really interested in is asking, at some level, if we have multiple genes, are we talking about 500 different diseases? Or can we coalesce this into maybe 10 or 15 pathways that we could then study? Do they come together somewhere? And so the big question, at some level, from a developmental standpoint, is when and where do autism genes act? And this is just showing brain development from the embryo through postnatal life. And it's pretty clear now from a number of studies, including our own, but I would say over a dozen studies have made it very clear that the autism genes that are being identified now are acting in this very early, mostly prenatal time, during the birth of the neurons, the cells in the brain, during their migration to their place in the brain, the things that make the connections, and which are called synapses. So the brain is very unusual in that the cells are born far from where they're actually going to reside, and in the human brain migrate over thousands of their own. The cell is 10 microns, and it's migrating millimeters and millimeters in the brain as it's growing. So this is a very complex process, but we've kind of pinpointed that most of the action is happening here. And so this is fortunate because we can use these cell models, which I'll describe in a second, to study how these genes might act. And so this is just a pictogram from 2009 showing that you can take mutations and put them in cell or animal models, and that you get disease insight because of mechanism. And then you can go back to even patient cells, and it's kind of a dialectic in a way, a back and forth between studying the cells, the patient, the animal model. This is another way of thinking about it. Where do these fit in our research armamentarium? So if we think about things, human relevance, of course, the most human relevant thing that we can study are ourselves, our own, our patients and ourselves. But the problem is it's very difficult to do the kind of experiments that give us true mechanism. We can't really take humans apart. We can't take our patients apart. We can't look at them as carefully as we can an animal model, and that's the critical part of animal models. However, we can't look at too many genes in animal models. It's a slow process. We have this notion of throughput, which talks about looking at, if we're talking about 500 genes, the ability to look at large numbers of genes very rapidly. And that's where the kind of induced pluripotent cells from skin, this is a stem cell that's made from a skin, or primary human stem cells from fetuses are actually grown. And I'm going to be talking about work mostly in these cells. These, again, if you can look at this and kind of see in 3 dimensions, which I'm really bad at drawing this, and I apologize for this figure now. You can see that these are the most human relevant, give you the highest mechanistic insight and the highest throughput. So for a disorder where we're going to have to look at hundreds of genes, or at least dozens, this really gives us a brand new kind of weapon to use. And so these are the 2 guys in my laboratory, both postdoctoral fellows. And Luis de la Torre Ubeta, who came from Harvard before here, is one of these CIRM postdoctoral fellows who's being funded as part of his career as well as Jason Stein, another postdoc fellow in the lab. And essentially, we get donors and we dissociate the brain in the dish, let's say a 12 or 15-week-old brain, and we can purify out the stem cells and differentiate them in these dishes, and then we can study them. We can see over a 12-week period how they differentiate and how they grow, and we can measure all the genes in the genome and how they're turned on and off with modern technology called microarrays or RNA sequencing. That's essentially what we're able to do. So we're able to grow these neurons. And it turns out that over 12 weeks, if I take let's say a 14-week embryo stem cell and grow it, it will recapitulate a lot of the same gene programs, we've shown this now, that are expressed up to about 24 weeks of gestation, 24 to 28 weeks. And so what we've done is we've compared what genes are turned on and off in multiple brain regions as part of this BrainSpan project. This is actually human fetal brain, and compared what happens in the dish. That's just a prelude to this. That's not the study, but what we know now is that our cells recapitulate this process quite remarkably well. And again, as I mentioned in the beginning, this is the period up to about 24 weeks when where most of the autism genes that we've identified so far are acting. So this model is going to be very powerful. And this is just showing you a picture. This is what they might look at 2 weeks in a dish. And what I'm showing are the neurons, the nerve cells growing here, and they're labeled green. And the neural stem cells or progenitors are labeled red. And here at week 12, there are no more progenitors. It's just these maturing, growing neurons, as would happen in the in vivo brain. And this is a close-up just showing that we can look. This is where the synapses, the connections between neurons, actually occur on these filopodia, these areas called dendrite spines. But these are like huge trees with huge arbors, and we can grow these and characterize these in a dish. So the notion is now we're able, with genetic engineering, to put autism risk mutations into these primary neural progenitors and watch them differentiate and ask what happens during development. We can look at their morphology, that is, do they grow the right kind of trees and connections? And we can look and get this RNA transcriptional signature, that is, what genes are turned on and off. And this is really critical. It's like a fingerprint. It's like a pattern matching. And we can use this to actually screen databases for drugs that can theoretically reverse this pattern. And then the notion is, can we then take those drugs and use them on the cells and actually show a proof of principle that we've actually reversed that pattern, and in doing so made the nerve cells in a dish function more properly. And the last question we can kind of ask here, imagine we can pretty quickly now put 24 different autism causing or mutations in these cells and look at them individually and ask, do they come together anywhere? Do these profiles or do their morphology, the trees or the genes that are turned on and off, do they intersect anywhere? Do these 24 different mutations converge on a pathway that then we might be able to target with a particular drug? So I think that's the end of what I'm going to say, because I wanted to just give you a broad 30,000-foot overview and just... But I think we're now open for questions.
I'm going to stand here to help point. So people who have questions about this type of work, where we are.
Questions?
Does that mean that autism can be diagnosed prenatal?
Some forms. Right now, there are kind of major gene mutations that you could say hugely increase the risk for having autism or intellectual disability. or a bad neurodevelopmental syndrome, and I would say that's about 5 or 10% of the cases now, somewhere in that range.
You mentioned you got to the point where you were able to reproduce the progenitors, I guess the antecedents in a petri dish. You left out were you able to reverse any of them?
Reverse in terms of
the action of the genes.
of the gene. Well, we haven't gotten there yet.
That was okay.
Yeah, because we spent a lot of the first year showing that these cells really do, because believe it or not, nobody's actually shown this clearly, how well these cells mirror in vivo the actual human brain development. And so we spent quite a bit of time really nailing that to make, to know what we were looking at and to be confident in our system that we're using before jumping ahead into it. So that's kind of where we are right now. We've made a lot of these mutations, we're growing them up and characterizing the cells, beginning to look at them both in terms of how the trees are growing out and how they're connecting to each other, as well as how the genes are expressed.
Is there any indication that there's outside influences, temperature or atmosphere or something else that might be relating to these things? Yeah.
Well, just, in general, in terms of causes of autism, we know that a large number of perinatal traumas, birth hypoxia, certain kinds of infections that occur like rubella, toxic exposures like valproate, which is a medicine but shouldn't be given to pregnant mothers. Those things can increase they're not causal, but they increase the incidence, the incidence of autism. So there clearly are environmental factors. The issue is that the genetics gives us a really great causal anchor, a place to start from. And I think one of the next, once we get past this and can show that we can model this, identify signatures, and maybe even use drugs to reverse them, a kind of next part of the field will be understanding how environmental factors can reverse them. For example, we can even take children and teach them language, many nonverbal children who are autistic. So clearly the environment, the brain is an organism that has enormous interaction with the environment. So there clearly is a role there. But what we're looking at here are kind of major genes. They're things like Fragile X kind of genes. They're things that have a really big effect. The environment might modify them a little bit, but it doesn't change the eventual outcome that much.
So, we're going to have time for questions as well afterwards during the social time. So,
I thought that many cases of autism had superior intellectual ability. Your diagram seemed to show inferior intellectual abilities.
Yeah, what I was showing there, thanks for that question, so I can clarify that. About a third, if you see those circles overlap, about a third have intellectual disability, but about 2/3 don't. So it's not a necessary part. In fact, many have, there's no IQ implied by having autism, but about a third will have intellectual disability. And a lot of it is due to these very more serious mutations like the Fragile X, tuberous sclerosis, and other new genes that we've recently found. So it's only about a third. So should I? Thank you.
Let's thank Dr. Coming up right away. Okay. Sorry, I don't mean to cut off questions because questions is what drives good science, but I also have to make sure we get out of here on time or they'll charge us more rent for the room. So. Is Eugene Washington here yet? Because he runs the hospital. Okay. Sorry, Dean. You'll get your turn later. So it's my pleasure to introduce Don Kohn, my close colleague in the Stem Cell Center. He's one of our senior members and really become a leader in various areas of genetic medicine here at UCLA. He has such a large number of titles, I'll just buzz through them. He's director of the Human Gene Medicine Program. He's a member of the Jonsson Comprehensive Cancer Center. He's a faculty member in the microbiology, immunology, molecular genetics, and pediatrics departments. And all of these credentials really speak to the breadth of the types of work he does, which goes really from the very basic all the way to the clinically applied, as you'll hear tonight. He's also been a wonderful leader coming to UCLA from Children's Hospital in 2009, and his work has really been groundbreaking. I'm sure everybody here has heard about the bubble boy, a severe immune deficiency where children with that disease had to live in basically a sterile environment. And he is one of the world's leaders in a genetic correction of that disease. And now there are children who have been treated that are living essentially normal lives outside of the bubble. With that type of track record behind him, he's taken on some new challenges, including diseases of the blood system that causes conditions such as sickle cell disease. So I'm delighted he's here at UCLA and very happy he's here tonight to present.
Well, thank you, Owen. I'm delighted to be here. I'm delighted you're all here. As Owen said, most of my career I've worked on bubble baby disease, and CIRM really gave me the unique opportunity to spread out and approach a disease that's probably 100 times more common, which is sickle cell disease. So that's what I'll talk about tonight. So this is the bloodstream, and so, or it's the 405 on a Friday afternoon. And so, as you know, blood is basically a liquid with cells circulating in it, and sickle cell disease affects the red blood cells. And what we know is that a type of adult stem cell, shown on the left, the hematopoietic stem cell, is the cell that gives rise to all of our blood cells. So on your right are all the mature cells that we see in circulation, including the red blood cells. And so in the bone marrow space in postnatal life, in circulation for babies in their cord blood, and the type of stem cells we can chase from the bone marrow into the bloodstream, mobilized stem cells, these cells for your whole life divide and specialize to become the different types of white blood cells, red cells, platelets, or the immune cells. And one of the clinical practices in my clinical work is in the field of bone marrow transplant, which is really stem cell transplant. And so in a bone marrow transplant, which is typically done for a patient with a bad type of leukemia or cancer to allow them to be given high doses of chemotherapy, we can rescue them from that state by giving them new stem cells. And so that's shown here. Oops. There's a patient under the drapes there, face down, and two doctors are withdrawing bone marrow from the back of their pelvic bone. And we can get enough bone marrow from one donor to treat another patient. And so the bone marrow may be taken. In our case, we process it in the laboratory to fix the gene defect. Then the patient would get the chemotherapy either to kill their leukemia, or in our case, just to make space for the cells to grow. And then the cells are given back. And actually, the easiest part of it in some ways is the actual transplant, because you can just put the cells into the bloodstream, which will take them through blood vessels into the bone marrow, where they can then start growing and making blood cells. So really the hard part is the medical care of the patient for the first month or so afterwards while they're recovering from this therapy. But if it goes well, these stem cells, either from a healthy donor or that we've taken from the patient and fixed, will take over all their blood cell production for the rest of their life. And so the disease that we are now working on is sickle cell disease. And we kind of joined this battle somewhat late, and probably would have been very difficult to compete for NIH funding that really tends to reward the first and not maybe the best, or at least somebody who's going to get it done. But CIRM really allowed us the opportunity to do that. And so just a little background on sickle cell disease. So it's a disease that we all know about to some extent, but in fact it's still very much a common disease that's still causing a lot of problems. So about 90,000 people in the U.S. Have sickle cell disease, and it disproportionately affects minorities, with about 1 in 500 African American and 1 in 36,000 Hispanic Americans affected by sickle cell disease. And it causes a whole variety of severe medical complications, recurrent crises of pain, acute chest syndrome, and ultimately organ failure in the spleen, the kidneys, strokes, etc. And so despite advances in medical care over the last several decades, sickle cell disease in California still leads to early mortality, with the average lifespan only about 40 years. And so this shows just in the children's perspective of it. They tend to be shorter, they have hours of pain, swelling in their hands and feet, and other problems. So this is the most complicated slide that I'll show. So this is how sickle cell occurs on one slide. And so in red blood cells, there is a high concentration of a protein, hemoglobin. That's what carries oxygen. And it's made of two protein chains, two beta chains and two alpha chains. The sickle cell disease, unlike what Dr. Geschwind was talking about with multiple genes causing the disease, sickle cell, it's one gene, and in fact, every person with sickle cell has the exact same mutation. So in the beta chain of the globin, the 6th amino acid is changed from a glutamine to a valine. And so just one base is changed out of the 3 billion in the base pair. That different amino acid winds up being on the surface of the hemoglobin protein, and it can actually connect to the next protein over. So that leads to all the proteins sticking together or aggregating. And it turns the red blood cell from a very flexible, soft donut shape to this very rigid sickle-shaped cell, and those get stuck in the blood vessel. And so sickle cell is basically vaso-occlusion, blocking of the blood vessel by these rigid red blood cells. So it's really amazing that from one base pair change you get these very complex clinical complications. And so of the 90,000 or so people in the U.S. who are affected by sickle cell, only about 500 have had bone marrow transplants, so about 0.4%. This was actually first done in 1976 for a patient who had sickle cell disease who also developed leukemia and needed a bone marrow transplant for his leukemia. He had the transplant, and it not only cured his leukemia, it also eradicated his sickle cell disease. So it really proved the principle. And of these 500 or so transplants, when successful, the results are no more painful crises, no need for transfusions, ongoing organ damage stops, and patients can really have normal activity. Of course, it won't correct pre-existing problems. That'll take some of the other stem cell therapies that we're working on to fix the brain, the kidneys, et cetera, that have been damaged. But what's really limited to such a small percentage are the immune problems. So we need to find a perfect match to give the bone marrow, and even with that, it's possible that a patient with a healthy immune system can reject the marrow, or the flip side, the bone marrow itself can reject the patient. That's called graft versus host disease. As a clinical transplanter, that's what we deal with. Our major problem is this immune complication. And so this is where gene therapy gets to work to try and find a different approach to the disease. And so sort of our overarching hypothesis for our work is that for these genetic blood cell diseases, fixing the gene in the patient's own bone marrow stem cells, which are then transplanted back to them, could correct the problem. So that each patient can be their own marrow and they're all perfectly matched. And so if we can do this, we think this will be a widely applicable approach for sickle cell and other blood diseases. And so simply the idea is to, going back to this chart, to get the gene in the stem cell to be normal. Then if that cell really is permanent for the patient for the rest of their life, they'll make normal red blood cells. And so the way we do it is we actually use viruses as our gene delivery vehicle. So that's shown here. This is meant to be a cell with its nucleus and DNA. We can actually clone into a virus a normal hemoglobin gene and use the virus to bring that genetic information into the cell and lock it into the chromosome. So now this cell is permanently modified to have this new gene, in this case a normal hemoglobin gene, that can be passed on to all the blood cells that are made and can make its therapeutic protein. So that's sort of the gene therapy technique to do this. And so what we've been working on under this CIRM grant for the last now 3.5 years is to bring it sort of from that idea to the clinic. And we're rounding the home stretch, I guess we'd say. So the idea is we would take bone marrow from a patient with sickle cell disease, in the laboratory isolate their stem cells, and then use a virus to add a normal hemoglobin gene that prevents the red cells from sickling and freezing the cells away. And then when we know we have a good cell product, bringing the patient, giving them chemotherapy to make space, and transplanting them back with their own bone marrow. And so over the past 3 years under our initial CIRM grant, we've done all the preclinical work to show proof of principle and efficacy. And so we just published a paper for scientists. This is one of our good goals. So we published a paper in the Journal of Clinical Investigation in which we just described basically 25 samples of bone marrow from volunteers with sickle cell disease, where we took their stem cells, added this gene, and in the laboratory turned them into red blood cells and showed that it prevented the cells from sickling. And this was funded by one of those CIRM disease team grants that you heard about. And so under this grant, we've also now developed the clinical protocols. We've developed what would be considered a phase 1 or first in human clinical trial. And our goals are to look at the safety of doing this, the feasibility and the efficacy of doing this stem cell gene therapy for patients with sickle cell disease. So as I said, it will use the patient's own stem cells modified by adding this human beta globin gene engineered to prevent sickling. And what we plan for the study is just a small pilot study for the first phase 1. A total of 6 subjects will be treated. And as we planned the protocol, we thought that it should be adults for the first patients and ones who don't have an available matched sibling or unrelated donor. And then we have a whole series of very precise criteria. We want people to be pretty sick but not too sick to make it through the transplant. And so we've defined that working with a team of hematologists both here and at Children's of Los Angeles and Children's of Oakland. And so where we do this is in our clean room. And as it says, your clean room is not like our clean room. Our clean room is like this. And so the Cancer Center and the Stem Cell Center and the School of Medicine support what's called a good manufacturing practice or GMP facility, which is suitable for taking cells from patients and doing these ex vivo or in the laboratory manipulations in an environment that's so pristine that the cells are safe to give back to the patient. And so we've now been sort of in the last year of the grant really doing the scale up and going from the laboratory small scale to doing things on a clinical scale in preparation for the trial. So these are our future steps that we hope to get done over the next 6 months to open the trial. And so we're completing our studies and developing all the basic procedures, both for the laboratory cell processing as well as for the clinical treatment of the patients. We've applied for another disease team grant for the actual clinical trial, and that's under review currently. And our goal for the first grant was to submit an investigational new drug or IND application to the Food and Drug Administration by the start of next year to be able to open the trial. And then once we know we're going, that we're funding and that we're approved by the FDA, we will then get the actual clinical grade vector to transfer the virus to deliver the gene, the other reagents. We'll then train the laboratory staff and the regulatory investigators, and we're aiming to open the trial next summer. So I will stop there and just thank, this is my lab, these are all the people in front of the Terasaki building who do all the work that I talked about. So thank you very much.
Thank you. Great discussion. Are you in the era where you have to find a naturally occurring virus to do your injection into the cell, or can you start to manufacture a virus-like entity to do this?
So I didn't say this, but actually the virus we're using is based on the HIV/AIDS virus. And so it's been cloned in the laboratory, and all the virus genes are removed, and just a human globin gene is put into it. And so it can carry that into the cell once and put that gene in, and it won't replicate. And so this technology of using these viruses has been developed over the last decade or so. There have probably been 24 patients with other diseases treated, and it hasn't shown any problems, but that's part of the safety that we'll be looking for.
Thank you.
Once you're successful curing sickle cell disease, would any of these techniques be applicable to attacking thalassemia or other hemoglobinopathies?
Yes, so thalassemia is another red cell disease where you don't make enough hemoglobin, so it's a severe anemia. This would be directly translatable to this. So the same approach should work exactly. We're just starting with sickle cell disease.
When you do the chemotherapy to, like you said, make space, is it also necessary for you to kill the stem cells, all of the stem cells that are producing sickle cell, or is there any worry that when you put in these new stem cells that you're going to be producing your good ones, but then still ones that can clog down the road?
Very good question. So the dose of chemotherapy that we plan to use should get rid of about 80 to 90% of the patient's own stem cells. And the red cells that they make that sickle have a very short lifespan. It's part of their problem. It leads to the anemia. Whereas the ones made with our gene in them will have a normal lifespan. So a small percent of residual ones shouldn't be a problem. There's been a few patients who have been transplanted that only got a small percent of donor cells growing, and yet it's helped them clinically. So we don't need to get 100%.
You mentioned looking at what I think of as kind of tween generation of people who with sickle cell, the young adults. Who determines the protocol or how quote unquote healthy they are in order to be part of the trials, or how do you see that working? Would you guys have a standardized protocol, or is it just kind of like they're doing sometimes by need?
No, so it's in the field of bone marrow transplant in general, we have very precise criteria measuring heart function, kidney function, lung function for who is qualified or not. So we have those types of criteria. And so before the transplant, there would be a series of tests, an EKG, an echocardiogram, chest x-ray, et cetera, to make sure that basically it's adequate organ function to make it through it, absence of infections and other things. So we have they're relatively standard for the field of transplant.
Dr. Kohn again. One of the things that may not be obvious to people who aren't involved in science on a day-to-day basis is just how incredibly fast these fields are moving, particularly with the advent of new technologies. Just thinking what Dan presented in his talk about microarrays and RNA sequencing, cell culture methodologies that are only within the last few years are being placed into his work. For Don, the advent of these lentiviral vectors, many labs working on that probably for about a decade now, but they have now become, in a sense, a tool that one can use in a sense, almost routinely to explore these kinds of clinical questions. There's still a lot to be done, but to my mind, looking at science, sitting here and listening to the survey talk, in a sense, it's an incredible amount of basic science that has to go into making these clinical events happen. And I think our next speaker, Antoni Ribas, my close colleague and friend here at UCLA in the Department of Oncology, his work also exemplifies that, taking the fruits of the basic science and applying it in our understanding of how to use immunology to fight cancer. Tony is a professor of medicine oncology, as I mentioned, and he's really a superb translational researcher. He's the person who takes these ideas, figures out the clinical trials, and takes them into cancer patients to try to change their lives and their outcomes. He runs a busy clinical practice, and he runs a research lab, and he also runs the clinical trials that you'll be hearing about. He has this incredible energy to get it all done and do it very gracefully. So please welcome Antoni Ribas.
Thank you. I'm here to talk about a group effort. I'm just the speaking head of this large group of people that includes Owen Witte, includes Don Kohn, includes Jimmy Conomo, David Baltimore, a series of professors and investigators from UCLA, from Caltech, and from USC that tried to merge expertise to be able to fight one particular cancer. And I was lucky that they will be focused on the cancer that I work on, which is a cancer called melanoma. That's a skin cancer, the most aggressive one of them, that until recently there were no good treatments for. Whenever somebody had this cancer, there was no therapies that were active. But we've known for a long time that we could get some patients to respond by giving specific therapies that turn on the immune system. And once this would happen, the cancer would go away. And whenever we would look inside what's going on in the cancer, we would see it was full of lymphocytes, of the white cells of the immune system, the white cells of the blood that are infiltrating and attacking the cancer cells. They're staining brown here, and the few cancer cells that are left are the ones that are staining in blue. The cells that made it there didn't make it just by chance. They were driven by a specific receptor that they have, which is called the T-cell receptor, that recognizes a protein that's on the surface of the cancer cell that's called a tumor antigen. This is one of the most elaborate receptor to ligand interactions that nature has developed, where if you just change one amino acid here, one little part of the protein, this will not work anymore. It's what evolution has developed so we can protect ourselves from the environment. It doesn't work that well to treat cancer, so we have to do something about it, to tweak it, to make it work to treat cancer. But we know that what gives the specificity for recognition is these two proteins called the T-cell receptor. One of the proteins called the alpha chain, the other one is called the beta chain. And they come from two genes. So as you've heard from Don Kohn, we've learned over the years that we can take genes from one cell, put them in a gene transfer vector, infect another cell, and then we have redirected this cell, and now we give these receptors that were able to attack melanoma from a patient where it all worked to a patient where it's not working, and then we can change the immune system of that patient. We can put it in a bag and shoot it back to the patient. So we thought that we could do this, but we didn't know we could do this, and we had to learn from several people. And some years ago, I think here you have 2006, we were called by David Baltimore, who was at that time the president of Caltech and a Nobel laureate, and with Owen Witte and Jimmy Kahneman said, we have this idea that we want to take this to the clinic, and let's build the team that can do this. And yes, we built the team. We keep meeting from time to time, and here some years later, and the team is bigger, and then we meet again, and the team is even bigger. Because we needed all these people to take things from the lab, from the idea, to treat patients. And the way we can do it is by having these premises in this campus, in the factory building, which is where I have my laboratory, where we can make cells and we can give them to patients. What Don Kohn was talking about, the clean rooms, where we have to gown up and everything is controlled. And we do this so we can get an army of immune system cells that we have genetically programmed to attack the cancer. And I can tell you a lot about this. I can show you graphs and things, but a mini movie is actually a much better way to explain it. We're trying to get this white cell, a lymphocyte, to attack this red cell, a melanoma cell. And I just put them like this because we just color code them. We gave them dyes and we put them in a flask. But the key of this is that we took the white cell and we gave it the receptor that allows it to recognize the cancer cell. Otherwise, the white cell will not be attacking anything. And then we put them together in a flask. On the bottom, there's a whole bunch of cancer cells in red. So the bad guys are in red. The good guys are the ones that we have genetically modified to attack the cancer, which are the killer lymphocytes. And we put it together, and we stick it in an incubator, which is a machine that keeps the human body temperature. We keep it there for 6 hours. But I'm just going to run it in 23 seconds. Let's see if it works. Here it is. Wait. There we go. So you see, the green cells, the good guys, are rounding up the red cells. They just look for them, and they start attacking them. Some of them just go wild. Look at them. There's one here. They just kick it. You're a bad guy. Well, this is what we want to do in humans. We can do this in a petri dish. We can do it in a flask. Can we do the same in a human? And at the end, you basically see that most of the bad guys are gone. They're clumped up. These are mostly dead cells. Well, we can actually do the same in humans. We can get the same kind of cells, manufacture them to FDA standards, and give them to a patient. And usually we start with lymphocytes that do not have the right receptors. If they had the right receptors, they would be in this gate. This is something called flow cytometry, where we look for lymphocytes that have a specific receptor that recognizes a protein in melanoma. So we start with this, and no wonder a patient is sick. But we can then change it. And we have the population of cells that were here. Each one of the dots was a cell. There you could barely see them here. Now it makes a big population. It's full of dots, and they're all up here. We have genetically programmed them to recognize melanoma. And we do this with this gene transfer technique. We put 1 billion one of these cells in a bag, and we give them to a patient, analogous to what Dr. Kohn was saying of a bone marrow transplant. And with this, we can change the immune system of the patient. We started with no cells here, and then we take blood of this patient, and now they look like the ones we have genetically modified. We've given through the vein. They have expanded through the body, and now they show up in the blood. And most of the cells, the white cells in the white lymphocytes in the blood, are now specific for the melanoma. Melanoma is a cancer that comes from pigmented cells. We have them all over our body. So actually, well, the receptors we gave to these cells are recognizing a pigmented protein. So one of the side effects is that patients lose pigment. They go from this patient being the same human being, just separated by 5 months, and this process where lost all of the pigment in the hair and in the skin. But we do this not to turn people all white, but to be able to treat melanoma. Here we have PET scans, which is an imaging technique where it makes the cancer cells glow. The PET scan always lights up in the brain, and it always lights up in the bladder, so this is not melanoma. Sometimes it lights up in the heart where the tracer goes around and in the kidneys. But the rest of these dots around here, all of these here, are melanoma. You can see there's a lot more here than here. There's these big ones here that are not here. This is the trace in the heart, so what's left is back here, or a whole bunch of lesions here that are gone here, that are decreased in number here. We could get the majority of patients to respond by giving this army of genetically engineered T cells that we shoot to the patients, and we change their immune system. Now it's attacking the cancer. We were really happy with this, but as we did it, we found that the cancers found a way to regrow. We wanted to study the process of that. It could be that the cancer had changed, and we studied it, and most of the cases it had not changed. But it could also be that the immune system cells that we gave that were killers in short-term assays, they didn't become as good killers afterwards. And to study this, there was no good technology to study it. Whenever we're using a lymphocyte or a T cell, a living body, as a treatment. It's different than using a chemical entity where you can follow over time. Here it's a cell that can change over time. So we had to develop new technologies, and we did it with a professor at Caltech, Jim Keith, and who was a graduate student, Chao Ma, who's now a doctor, after doing all of this research where we were able to select the cells, make microchambers so we could study them and study different functions. And we use words like microarrays, chips, microchips, and multifunctional, a whole bunch of complicated things to be able to understand the biology of these cells. And I'm not going to go through that. I'm going to just tell you what happened afterwards, what we learned from it. We learned that we give the cells, they expand in the blood, they repopulate, they reconstitute the blood of the patient. They're now good fighters. And with time, they start decreasing in number. But as they decrease in number, they decrease in their function. When Jim Keith, who's the Caltech professor, first told me about this, I said, yeah, I get it. But he saw that I wasn't getting it all the way. So he said, I'm going to explain it a little bit simpler. Tony, what you give to patients are like superheroes. You give these cells that are like Superman. They have a whole bunch of functions. They can do this and that and that. They can fly. They are strong and everything. But as you recover them from patients, they start being less of a superhero. They go down a little bit on the scale. They become a Batman. As you recover them a little bit further time, whenever the tumor starts to regress, they're not that much of a superhero anymore. They're like the sidekick to the superhero. They're the Robin guy. But the worst thing about this is that the cells that were left at the beginning, they were actually really lazy cells. They're cells that are not doing much. They're helping the bad guys. They're becoming like Homer Simpson. Well, we want to understand this so we can reverse it, so we can change it. And we thought that we could change it by giving stem cells. Why would that be? Well, that's not my idea. That's an idea of David Baltimore that has been also tested by Jerry Zag and Don Kohn, where if you can put those genes that redirect the lymphocytes, not on the terminally differentiated lymphocytes, but on the mother cells, the ones that develop endogenously into the fighter cells, then we could have a continuous progeny of T cells that are able to fight the cancer continuously. So we're going to be doing this. We're behind Don Kohn's approach for probably a couple of years, but we're going to be giving patients the cells that we're giving now with the T cells, with the gene-modified T cell receptor. They're good at controlling the melanoma initially, but then we want to maintain it by putting the same genes into the stem cells and then allowing this endogenous army to be developing over time. And that, as Dr. Thomas and Don Kohn said, is funded by the California Stem Cell Initiative. It's a $20 million award. We haven't been able to start it for the last 3 or 4 months. It's been stuck with an intellectual property issue, but we're going to solve that because we want to bring this to patients, and our timeline is to be able to do this in 2 years in the clinic. So with this, I just wanted to say that we're able to organize a team of investigators who are focused on an idea, who are focused on bringing to patients new therapies. We can do it within the premises of an academic center and be able to bring the therapies to the patient, learn from them, and then step back and see what should we do now to be able to improve on this therapy. So I'll be happy to take any questions. I didn't explain it well enough, I think. No? Maybe it's too late.
In your first slide, you showed cells taken from a patient cured after 5 years. Is not that 5-year number arbitrary?
Oh, I didn't say cure, and I'm a medical oncologist. I'm very careful about using the word cure, although I think with some of these patients that are durable responders to immunotherapies for cancer are probably cured, even if they started with a metastatic cancer. That was showing that the patient was, the picture, what was happening 5 years later, which there was no cancer. I was trying to say that the cells that induced that were the ones that we wanted to study, and we learned what were the receptors that were able to do that and get a response that's that long. So I hope I didn't misspeak. I didn't try to say cure.
Other questions, Dr. Ribas?
Suppose for your modified lymphocytes treatment, I guess to suppress the cancer, you need multiple treatments instead of one time. So for your stem cell treatment, what's your expectation of efficacy?
Yes, it could be. We could think that we could do the short-term fix multiple times, and then we would get a better treatment. But the patients go 3/4 of a bone marrow transplant, and that's quite toxic. So it's something we cannot be doing continuously. That's why it's very attractive, the idea that we program the blood system, the hematopoietic systems, the stem cell, to be able to make the T cells all the time, and that's what we'll be trying to do. Then we'll just have to do it just one time.
Question back front.
What's the volume of bone marrow that's needed to transplant? Is it half a pint?
Why don't you repeat the question?
Yeah. So the question is, what's the volume of bone marrow that's needed to do this transplant? We actually take not bone marrow, but we take circulating stem cells. There's tricks to make them mobilize, go out from the bone marrow, go into the blood, and then they're trapped with a machine that's called, that's like a centrifuge that takes away the blood cells, just keeps the white cells and gives back the rest. You get it back like this. It's 300 milliliters, so I don't know what that in pints is. I'm originally European.
Tony's European. He doesn't do that metric English. Any other questions? One in the front.
Is the treatment once or is it
do you want to wait?
Oh, I'm sorry. You do the treatment one time, or it's followed up by some sort of booster for the immune system?
It's designed as being a one-time deal, where we change the immune system, we regenerate it to be cancer-fighting, and then we give it back. So it should be a one-time deal.
Thank you, Tony. Well, to finish up the evening, it's my pleasure to introduce Eugene Washington, who is our Dean of the School of Medicine and Vice Chancellor of Medical Sciences here at UCLA. We're sort of in his house. The Ronald Reagan Hospital is part of the things he runs. And a really pleasure for me to introduce him because I've gotten to know him as a friend and as someone who really cares about how clinical work impacts our society. He's a health policy scholar for many years and a well-known clinical investigator. And here he's got a ride herd over all of us. So just ask him to say a few words, and then following his remarks, we'll invite all of you outside for some refreshments. So, Gene.
Thank you, Owen. Well, Owen, I'll remind you, this is a public institution, so this is our house this evening. I spoke at a dinner on Saturday night in Washington where I was the last speaker, and so when I stood up, I could tell the audience was flagging a little bit. So I suggested to them, 9 o'clock East Coast time, that they pretend they were from California. It didn't work, so I cut my remarks short. But I don't want to add and can't add anything to this brilliant scientific presentations that have been made, but I would like to reinforce 3 points that have been made over the course of the evening. One is the point that John made, Thomas, at the beginning, and that is to say thank you to you, the citizens of California, for making this investment in science, which in turn is making an investment in the scientists and in the communities that we serve. So you should think of this as, for those of you who are involved, as a shareholders meeting. And we're sharing with you the fruits of your investment, and think of this as sort of a down payment. But there's more return on this investment to come. The second point I'd like to make, another one that's already been underscored, is that this is truly a revolutionary time in science and in medicine and in technology. We enjoy unprecedented understanding of human biology. At the same time, we have new tools and techniques that were unimaginable just 15 years ago. And as you heard, in the last 18 years in particular, last 8 years in particular, the Broad Stem Cell Research Center has been an active participant in this revolution in science and in technology. And one of the reasons, obviously it starts with the great leadership of the individuals involved, but it's also been our ability to recruit the best and the brightest. There was an allusion to the great young scientists that we have around, and we do have quite a bit. But it's the staff also that we've been able to recruit, and it's the trainees at multiple levels, from students to residents, and particularly our graduate students and our postdocs. But even with the great leadership, obviously the vision, and with these great individuals, we wouldn't be where we are here at UCLA or in stem cell and science without the tremendous partners. What was noteworthy about one of the slides was one of the ones that Don Kohn put up. If you looked at the partners on there, you know, we have competitors here in Los Angeles, across town, but they are also our collaborators when it comes to advancing science and ultimately delivering on this promise of stem cell in the form of treatments. And so if you looked at that slide, it had USC as a partner, Caltech, and not just academic institutions, it's also partnerships with industry, which is very, very important as we think about translational. So that what I'm particularly proud about in the stem cell is that for us, in many ways, or for me, it's an exemplar of what we want to achieve in big science, because it really does promote and nurture the collaborative spirit that's necessary for real innovation and high impact ultimately as a result of these multidisciplinary endeavors. And then the final point I would make is one that's also been made, is that the future truly is bright. Again, going back to John's comments, big stuff is ahead down the road. And I can tell you much of that big stuff is going to be unfolding right here at UCLA. Owen and I share the vision that it's not enough, and this is along with our other colleagues in the Stem Cell Research Center, it's not enough for us to deliver on the science in terms of publications and in terms of these great discoveries where we unlock some of nature's best kept secrets. And it's actually not enough for us to really advance the translation, as we are beginning to do in the form of clinical studies. The ultimate goal is eventually having treatments that are available in the form of cellular therapeutics. And we intend that we will be right at the forefront in terms of leading this next phase of the revolution, and in turn, not only yielding a high return on your investment as citizens of California, but ultimately leading the country and the nation in improving the lives of individuals who can benefit from these cellular therapies. So I want to congratulate my colleagues in our Stem Cell Center. for the tremendous work that you have done and the contributions that you have made and the way that you serve as an example for all that we want to achieve across our UCLA health sciences. And I want to thank you for inviting me to participate in the program. I really enjoyed it very, very much, and believe me, I've learned a great deal this evening as well. And so I'm going to ask our audience to join me in a round of applause for all of our presenters this evening.